Buenos días. Vamos a iniciar la jornada de hoy sobre el tema de Huntington disease.
No sé si lo tiene.
Bueno, y entonces...
No lo tiene y...
Ah, ¿lo tienes? Bueno, esto del directo... Bueno, ¿me oís bien? Vale, pues sigo. Entonces, quiero agradecer a Bancaja la cesión de local, a todos ustedes o vosotros o vosotras la asistencia y a los colaboradores y participantes el magnífico programa que han preparado para nuestra mayor ilustración e información. Sin más, doy paso a la doctora López.
Buenos días. Good morning, everyone. First of all, I would like to thank you, la Fundación Valenciana de Estudios Avanzados, for the collaboration in organizing this joint initiative between the University of Valencia and the Third Cardinal Herrera University. This initiative aims to raise awareness among health science students about these rare diseases that progressively deteriorate, as well as to raise awareness in a more unsupportive and cruel society about the difficulties faced by people affected by this disease. I would especially like to thank Javier Quesada, Secretary of the Foundation and Executive President of the Jaime I Awards, for his kindness in organizing so many health events, even though health science are not his specialty, because he holds a degree in economics from the University of Valencia, a PhD in economics from the University of Cincinnati in Ohio, and is a professor of economic analysis at the University of Valencia. Thank you very much for demonstrating your neutrality towards universities, as all have place within the foundation. I will also like to thank on my own behalf and on behalf of Dr. Bataller, Dr. García de Yébenes, winner of the 2000 King Jaime I Prize for clinical medicine for his studies on neurological diseases and for his participation in this session, as well as Dr. Minichiello, Professor of Cellular and Molecular Neuroscience at the University of Oxford. Thank you, all of you, and welcome to this great event that is going to be now. Okay, thank you very much.
Professor Bataller.
I just want to add thank you for the invitation. Thank you for this opportunity to share the knowledge. And I think this session is very interesting for all the students here because it mixes people speaking. Dr. Justo García de Yébenes, who is a very, very well-known clinician in neurological diseases. We've all have learned a lot from him. And also Dr. Minichiello, who is a basic researcher devoted to this area. I think that this friendship or this collaboration between clinicals and basic scientists is what makes this area go forward. Well, thank you.
¿Está en la sala algún representante de la asociación? ¿Corea Huntington? ¿No? Pues nada, entonces tiene la palabra doctor Javier Quesada.
Good morning, everyone. As it was said, I'm the president of the foundation, executive president of the Foundation Rei Jaume I. We do two things, essentially. During the year, we organize, as today, activities related with science, innovation, and technology. But our main purpose is to organize also annually our well-known Rei Jaume I prizes. We are in our 35th edition. This year's is the 36th edition, so we started with these activities under the leadership of Santiago Grisolia, a well-known biochemist, a Spaniard who lived in the United States for more than 30 years and then came to Spain and brought this idea of collaboration between companies and universities and scientists to really teach the society how important is science and technology for the welfare of the society. And well, he passed away 3 years ago, at the age of 99. And he was active until the end of his life. And he had this vision of trying to modernize Spain also in this sense, of bringing these two worlds, corporations and scientists and universities, closer together, because this is still a subject in which Spain is still very delayed. So we organize annually these prizes. We bring to Valencia every year around 22 Nobel laureates, which is something that doesn't take place anywhere else in the world. And we are very proud of keeping them coming back, which is really the honor for us that they consider our prizes very well organized, but also very independent prizes. We have an example here of Justo García de Yébenes, who won this prize 25 years ago, and he's still active. Of course, he's in neuroscience, so he knows how to keep himself in good shape. We bring them in June, so in a couple of months you'll see that in Valencia there is like 2 days of activities related with science. And then in November, usually the royal house, the king or the king and the queen come to Valencia to deliver the prizes. And we organize many things. So if you enter our website, you'll see that there is always activity dealing with science, technology, and also innovation. And we invite you to come to our activities, because our activities are well above the mean of the activities in the city. And if you go often to these activities, it's like reading a good newspaper. You'll get culture, which is based basically on many, like when you paint a house, you don't use a lot of paint. You have to paint 3-4 times, right, to make it solid. And I invite you to use our foundation. We are always fighting to try to bring public to our activities. And it's not an easy way. It's not an easy task. So I welcome you to this activity. And I declare this activity open, and maybe it's time to make a picture and get started. Thank you very much.
Thank you very much.
Vale.
Aquí arriba. Aquí arriba. Bueno. Thank you very much. La próxima vez es aquí, creo que prefiere que nos la hagamos aquí arriba la foto.
somebody to operate this so that I just get down to my presentation? Because I don't know if it seems. My presentation is below this one, I suppose. Yeah.
But in the same.
Yeah.
In the same.
No. This is another presentation. So this should go away. Okay. And then I should find myself. And okay, should be this one somewhere. I mean, the presentation seems. This one seems on. I just need to get to it. Okay. Oh my God. I mean, it's really terrible. Let's see on top. If we go on top here. There. So where is?
But this is what it is here, in here.
No, that's not mine. It's not, no. That's why. Passport. No, wasn't that? We go out of this again? My passport? Yeah, I think it's this one. Let's see which one. Mine. So now if we go down. Where's mine? Oh my God. This is mine. Okay, how can I get to my... Yes, exactly. So I am actually in my passport.
Yes.
Okay, so if we go, should be down here.
Down.
This one?
Rei Jaume I.
Yes.
And open.
Yeah.
No, I don't know open.
Like that?
Yes.
Okay.
It's okay. It's okay.
Jeez, I mean, I don't know how
And
to
this
use.
one, and this one.
I don't know how to use them really.
It's okay. But this, okay, it's okay. And this,
Well, actually, I think
to pass, okay? Pointer. This is the pointer, and no. Okay. You can see the pointer here and here. Okay.
I can only see the arrow.
Okay. Oh, I don't know.
Well, thank you. We will start with the first, with the scientific session. It is my pleasure to introduce you to Dr. Liliana Minichiello. She is originally from Italy, and after developing a career in basic neuroscience in Italy, the United States, Germany, she's now settled in Oxford, at the University of Oxford, where she's professor of cellular and molecular neuroscience. She's an expert on the study of molecular and cellular mechanisms and underlying synaptic plasticity, learning, and memory. She's been working with very advanced techniques such as single-cell transcriptomics, and she's talking us about things that have to do with the Huntington's disease in the biology of neurons at the basal ganglia and the influence of brain-derived neurotrophic factor signaling in these neurological circuits. Please go on. Thank you for coming.
Thank you very much, really. I feel really honored to be here, and thank you for the invitation. This is really something fantastic. And what I appreciate most is actually what has just been said, that when actually one studies neurodegenerative disorders or any other type of disorder, I think the clinicians are what you need to work with, so that basic science and clinician, they work together, so you see what the patient look like. This is what I'm aiming for, because my science is mainly actually basic science. I'm trying to understand the mechanisms, how things happen. And this, I suppose, are very, very important too, because actually these are the ones that will help you to generate new therapies. But of course, then you need to see the patients, you need to understand how they suffer, and so on and so forth, to be able can you hear me? Yeah, that's good. Thank you. So to be able basically to apply a therapy of some sort, we really hope to manage that. So this particular presentation today in fact, is trying to understand mechanisms that can work, basically, in Huntington disease. And you will see that I call my presentation here Alternative Strategies for Neurodegenerative Disorders. You will understand why. Is because we don't use models directly that affect, basically, like mutant Huntington that affects, basically, the mouse. And then actually you are confounded by all the problems that this carries. But we try to isolate problems. And we are focused on the BDNF TrkB signaling in this particular occasion. But you will understand more while we go through. So the first thing that we ask ourselves, and I think this is a question everybody asks, is whether or not we can treat neurodegenerative diseases. And so at present, as you may know, there are no cures for any of this particular disorder. They are incurable, seems like. And so treatments that are available usually manage symptoms, but obviously, as you may know, they cannot halt or reverse it. So it is crucial that we understand how the degenerative process is triggered to then be able to control it. So because of that, I just give you like a few more lines of background to this. So how do we understand molecular basis of neurodegeneration then? So the main risk factor, as you may know, in neurodegenerative diseases such as Alzheimer's, Parkinson, Huntington, or amyotrophic lateral sclerosis is increasing age. So basically means that with age, it's more prone to get these particular disorders, even when they are genetic disorders such as Huntington. So most of the slowly progressive neurodegenerative diseases are characterized by similar events. And these events are, first, that each of these disorders seem to affect, in the first place, a particular subset of neurons, and then actually expands to the entire brain. Each of these disorders shows accumulation of aberrant or misfolded proteins. Another important point is synaptic failure, failure of axonal transport. So all of this basically makes the life of a person with the disorder really impact the life of these persons significantly. So there is a major problem, though, that we all find when studying the molecular basis that underlie the pathogenesis of neurodegenerative diseases. And this is exactly to distinguish the primary effect to these secondary effects that happens. So with this in mind, so we actually have devised our own approach. So we chose Huntington as a disorder because we know that that is a genetic disorder. So we know the gene, basically. But we focused basically our attention in trying to understand the mechanism that makes particular neurons that are first affected in Huntington prone to the disease, basically. And so for this reason, we are trying to understand the role of neurotrophin system in this process, and in particular BDNF and TrkB signaling. And this is because this function of this signaling system is believed to contribute to the neuronal degeneration in this disease. Okay, so next. This is actually the family of neurotrophin receptors. They are called TrkB receptors. There are mainly 3 of them, but the important one today is TrkB and BDNF, basically. So it's TrkB receptors that bind brain-derived neurotrophic factor. And this, we will look at the importance of this signaling, basically, in the basal ganglia, where the Huntington is first placed. So next. Obviously, I mean, you will have much more from Justo afterwards, but what is Huntington disease? So Huntington disease is a genetic, progressive neurodegenerative disorder of the basal ganglia and is caused by an expansion of the polyglutamine repeat at the N-terminus region of the gene Huntington. So the mutation that you can see here really consists of this particular addition of CAG repeats. Now, what are this? So basically means that the person that goes over the 35 repeats is prone to get this disorder. Now, what are the symptoms? Just briefly, so you have two types of symptoms: the locomotor and motor coordination related disorder, and these are involuntary movements like chorea, and muscle weakness, clumsiness, gait disturbances, among others. And cognitive disorders also occur in these people. And this actually leads to progressive severe dementia, irritability, anxiety, and depression. So the degeneration of in this particular disorder first initially occurs in a selective neurons, which they are basically placed in the striatum. Show you more in a sec. And they're called encephalinergic expressing neurons, striatal neurons. Now, if we go to the next slide, please. Don't freeze. Sorry. Let's see. Okay. I don't know what is happening. Next slide. Okay. So these neurons I was talking about are actually in the striatal region of the brain. So the principal component of the basal ganglia, thank you, yes. So the principal component of the basal ganglia is called striatum, and in humans, you see, is this region here in the brain. So in the striatum, actually, the major cellular component are called medium spiny neurons. Okay.
Because you are not.
I don't understand how. Sorry, guys. I mean, where is now the?
Why is not moving?
So maybe this should be placed like a presentation.
I know. Yeah, exactly. That's the point. We need to put. Yeah. Where is the mouse?
Well, it's not in a presentation mode, so then it means that it will always do that, basically. So if you have the cursor somewhere.
Yeah, but the problem is that we don't see the mouse.
Okay. So you just need to put that in the presentation mode.
No, it is in the presentation mode.
Okay.
No.
In the look.
Okay, bye-bye. It's okay. I think it was
No.
just...
No. Let's try to fix
No,
it.
but es que hay dos there's two screens.
Oh, that's
The
I
screen
understand.
there is okay.
Okay.
Oh, I see.
Well, it's not, because we don't correlate the two. Okay, wait a second. Let me just...
But it's
Okay, no. It's only to be with this, because otherwise it goes on the side. That's
Okay.
the actual reason, I think.
Okay.
But you see that there's like another screen here, which is what you are projecting.
Okay, and I am on this one now?
¿Me lo puedes arreglar?
No.
Porque es que no está en
Es que está
proyección.
como duplicada la pantalla. Lo que sale aquí no es su escritorio.
Sorry.
Lo ideal es, no sé, que ella vea lo mismo que está en la pantalla.
This is not my
Okay,
presentation.
it's open other.
Okay, then actually...
Guardar.
No guardar.
Guardar.
Ah, joder.
Porque era la del profesor.
Sí, no guardar
No,
cambios.
pero no
guardar
pasa nada.
cambios.
That is it, yeah.
Is this
Yes.
two presentations cargated and
Okay,
No, pero
we were...
ponlo en proyección, por si no, no se ve.
yes,
So at the moment it's not over there though.
No.
it's okay.
Okay, good. Okay,
It's
now
okay.
it's there.
Now it's there.
Now
Fantastic.
it's there or there.
Thank you.
It's the
Yes.
same, you see.
Sorry about this.
Fantastic.
Okay, guys, we're back. Sorry. Oh my God. Okay, so we were saying which ones are these neurons that are first affected in Huntington, and we said that these neurons are placed in the striatum, that in humans is this structure here, the neurons that we are talking about, they are called spiny projection neurons. Spiny projection neurons, they form like 90 to 95% of the striatum. So they are the most important neurons in the striatum, let's say. Now, the spiny projection neurons, though, depending on the target, they are divided in two subgroups. So you have the striatal pallidal neurons, which form the so-called indirect pathway, and you have the striatal nigral neurons that form the so-called direct pathway. Now, for the striatal pallidal neurons, they express enkephalin and the D2R receptor, whereas the other part express substance P and the D1 receptor. Now, beautiful. Okay, so what I was alluding to is the fact that these neurons are the first to go in Huntington, but this actually is not something we have established. This has been established on striatum from patients suffering from Huntington disease. In this particular slide, that is from 1988, so you have on this side two normal sections of a striatum where they express, to highlight the two pathways. So you look at the substance P that highlight the direct pathway and the enkephalin that instead highlights the indirect pathway, which is the one that is the first affected in Huntington. In fact, if you go to stage 2 and then stage 3, you see that they totally disappear, whereas the substance P, they are still present. So this is a fact that has been established in humans. So about Huntington then, so the function of Huntington has tons of functions, but among these actually, Huntington regulates also gene expression, and it happens to regulate the gene expression also for BDNF. This happens in the cerebral cortex. So BDNF is produced in the cerebral cortex, and mutant huntingtin participates to its expression, is then actually transported, where mutant huntingtin also helps, to the striatum. And in the striatum, on the postsynaptic site, you find track B, basically. And upon release of BDNF, track B binds BDNF, and in vesicles is then transported back to the striatal neurons, where it acts, basically. Okay, so because of this, there are basically two key cellular pathogenic mechanisms that have been highlighted, such as transcriptional dysregulation of BDNF and defects in vesicle transport of BDNF, which have implicated this particular signaling system in the pathogenesis of Huntington. So the point was to understand how BDNF track B signaling is involved. So the first thing that it was hypothesized is that BDNF track B signaling makes these neurons vulnerable by killing them. So basically that survival would have been the most important functions there. But we couldn't agree on that particular thing because, as I told you, BDNF is produced in the cerebral cortex, not in the striatum. And its receptor is in the striatum. So say that BDNF is affected either in expression or in transport, it means that in expression, if is affected in expression, means that affects other functions in the cerebral cortex. So we were wondering whether what actually was seen in the striatum was a result of this problem that BDNF has in the cortex, or actually was a direct effect through track B receptor. So how do we solve this problem was by making a specific and more pointed mouse model. So the mouse model we made was to affect its receptor in the striatum instead of actually affecting BDNF in the cortex. And for that, we just generated this particular Cre line that carries Cre under the pre-proenkephalin promoter, which suggests that when you cross this to a flox track B, you can delete track B specifically in these enkephalinergic cells, leaving intact, actually, track B in other cells of the striatum or the cortex. By doing that, actually, so the first thing we wanted to understand was whether or not there was a survival problem. And instead, we found that this was not the case. So if you take away BDNF track B signaling from these neurons, they don't die, actually, as such. And in fact, this actually just to show you that if you use a marker that looks at all medium spiny neurons, you find them all there in the striatum. What actually is starting to be affected is the expression of enkephalin in these neurons, even though the neurons are still alive. So, and this is known that if you reduce TrkB signaling, you affect the expression of enkephalin. So basically, what we show here is that the neurons are present, but they lose identity. And morphologically, as shown here, they're still intact. This was like a great surprise for us, since the field says that for 30 years that these neurons will die, basically. Okay, so then we were after understanding what is the function of these neurons, basically. I mean, what is the function to which TrkB contributes in these neurons? So it is known, actually, it was known, that if you basically kill these neurons, loss of these encephalinergic neurons give rise exactly to unwanted movements. So leads to hyperkinesia and chorea that is typically associated with Huntington. So basically we asked whether TrkB signaling would have such a function in these neurons. So here I'm just showing a few of the results that we got to show that, in fact, if you leave these mice for longer, so like when they reach like 8, 9 months of age, you see that what is affected is in fact motor behavior. So basically the mice show hyperlocomotor behavior, hyperactivity. So, and this one is just an image in the open field where this is the control at 9 months of age, and this is the mutant that actually run around the open field, rather significantly much compared to its control. So basically this data and other behavioral tests we conducted were telling us that the striatopallidal pathway, in absence of BDNF TrkB signaling, they lose inhibitory control over locomotor activity. So this was a good, sorry about this, go away. Okay, so I really don't know how to operate this guy today, sorry. I mean, I'm not doing anything in particular. Okay. Really, I have no idea what is wrong with you. First part, if you think about. Now, before we got ahead with the rest of the experiment, and because this is bioinformatic analysis, we wanted to be sure that this was true, actually, if you did an experiment. So basically, we wanted to test whether oxidative phosphorylation is truly affected, and so mitochondrial functions are affected. And for this, actually, so in this particular slide, it just tells you that the components of oxidative phosphorylation chain are mainly 4, and they are divided in 4 main complexes. That is complex 1, 2, 3, and 4. And all the electrons that come to this system, they come basically from DNA DH and the FADH2. Now, when actually how do we measure these complexes in our mutants and controls? So the way we measured that was to use an high-resolution respirometry device that would help us to analyze the different pathways in the mitochondria. And so the different complexes. Now, without making it too long, you can see that the pathway affected were the complex 1, basically, that is here. So this is the control situation at 3 months, and that's actually how this is affected in red. And the complex 2 and the combination of complex 1 and 2. Obviously, what this slide, even though it's a little complicated, also tells you that with age, normally in the control situation, the oxidative phosphorylation tends to go down. So imagine if you have a problem that on top of that affects these complexes. So basically, this was a true thing. So the TrkB and BDNF, in its absence, affects energy metabolism in these cells. So, but you know that early mitochondrial dysfunction and so energy metabolism and aging are the most common theme in many neurodegenerative disorders, including Huntington. So we actually asked, how is this dysfunction triggered then in the ISPN that lack TrkB signaling? So basically, we were looking for the trigger, actually, of this oxidative phosphorylation problem. So it is obviously known that excessive striatal dopamine can be a problem. Dopamine, it's been shown through the dopamine receptor 2 to affect, basically, to influence mitochondrial function and physiology, and to act synergistically also with the mutant huntingtin to induce neuronal death. So we were really looking for a problem that could have been caused by the absence of BDNF-TrkB signaling that had to do with dopamine. So what we knew was that in a rat model of mutant huntingtin, there is actually a biphasic way of dopamine dysfunction. So early on, around 11 months of age, this particular rat, as shown in this picture, shows an increase in dopamine, basically, in the striatal region, dorsal and ventral striatum, highlighted here. And this was known to be due to the fact that there is an increase, actually, in the dopaminergic cells in the substantia nigra in this rat, and also in the ventral tegmental area compared to control. And that's shown basically here. Okay, so then we were thinking, can it be that something of this kind happens in our model? Not only that, also in humans, is known, in human suffering from Huntington, that dopamine shows a biphasic way. So at time-dependent changes that they are initially, there is an increase in dopamine, like pre-symptomatic H0, HD1, to have then later on a decrease in dopamine. So the biphasic dopamine levels that we saw in the rat model were mimicking basically what occurs in human patients. Okay, so basically early studies in post-mortem brains of human patients have also shown increased level of dopamine and increased activity of tyrosine hydroxylase, which is the enzyme, the limiting enzyme in the synthesis of dopamine. And it has also been shown that dopamine depleting agents like tetrabenazine, actually, and DA receptor antagonists are able to reduce the abnormal movement in patients and demonstrating the contribution of dopamine to motor dysfunction, chorea, basically, in Huntington patients. So we looked in our model to find whether or not there was an increase in dopamine, and we did find that. So as shown here, we find that at 3 months of age, when actually there is all this problem with gene expression, we do find that there is an increase in dopamine, which later on, around 8 months of age, then decreases. So these are just Western blotting to show the same thing. And this is not due to the fact that the striatum, for instance, changes in volume. There is no problem in volume changes. So it's really due to the fact that there is an increase in dopamine. This is just to show you that if you do HPLC analysis, you see that there is the increase in dopamine and its metabolites at 3 months of age, 3.5, and then they go down basically around 8.9 months of age, really mimicking what is happening in the rats and possibly in human HD. And what was this due to? Obviously, we did counts of tyrosine hydroxylase dopaminergic cells in substantia nigra, and as in the rat model, we could show that this was due to an increase in cells that now express dopamine and brings dopamine to the striatum. So this was quite a big achievement for us, and because actually it was showing how BDNF-TrkB signaling can possibly affect these dopaminergic neurons and also the striatal neurons, the ISPN, that become possibly vulnerable because of the presence of dopamine for at least 3-4 months in the brain of these animals. Now, we were trying to understand what is the trigger of that. And as I showed you, without further ado, because otherwise it takes really too long, so we actually, let's say, analyzed the glutathione metabolism pathway, and in particular, this GSCO2 gene that was the most upregulated in that pathway. So GSCO2, actually, the function of this particular gene, the only function known, let's say, of this gene is recycling of vitamin C. And that was quite an important point because, as you may know, vitamin C is very important, apart to be very important for oxidative stress, but it's also taking part in the synthesis of dopamine. And so this actually illuminated us in a way that we hypothesized that possibly is because of this gene that is actually upregulated in absence of TrkB signaling, that actually you have more vitamin C recycled that goes basically to the substantia nigra and activates neurons, which are prone to express now tyrosine hydroxylase. And so basically, this is what this slide should show you. First of all, we tried to understand whether GST02, actually this is an SM fish analysis, an mRNA analysis, that shows that the striatal neurons expressing enkephalin have an increase basically of GST02 around 2 months of age already, whereas tyrosine hydroxylase is still not upregulated at that stage, but actually takes a month before then it becomes upregulated. And in this one here, it's just HPLC analysis to show you that we do have more of vitamin C that from the striatum goes to the substantia nigra, it's upregulated there, together with the transporter of vitamin C. So this was really suggesting that possibly what we have postulated was true. But how do we make sure? So basically, we decided to downregulate the expression of GST02 in brains of mutants at 3 months of age. So if that was true, so if you downregulate GST02, you should basically level dopamine or reverse dopamine increase, and you should also avoid hyperlocomotor behavior in these mice with time. And this is exactly what we got. So this is just to show you the constructs we made. We intracranially injected these mice with construct shRNA that would downregulate GST02, and then we tested these mice at different times, and we can show that now the locomotor behavior has been normalized, whereas mutants which are injected with scramble shRNA, they still show their problem, actually, and even when we reach 12 months of age, the rescued mice, they still don't show the problem, suggesting that possibly we have been able to rescue this problem in these mice. So the question then was, do we rescue also early motor behavior in these mice? So we know that in these mice, the unsupported rearing, that is when the mouse stands on its feet, basically in the middle of the cage, is affected in our mice. But when actually you rescue with GST02, then basically these also are rescued. Okay, so then actually the next question was to understand whether or not dopamine was actually rescued. And just not to make it very long, yes, it's true. So basically at 3 months of age, we have also rescued the dopamine levels. The cells in the substantia nigra now, they are basically normalized, basically both the substantia nigra and the VTA compared to the normal mutant, and this is shown here by counts of these cells. So GST02 upregulation, basically, and the hyperdopaminergic state that has been observed early at 3 months in our mutants interact to actually produce this hyperlocomotor behavior. And obviously, you want to understand whether you also have rescued the energy metabolism. And this particular slide here suggests that we are also basically able to rescue mitochondrial dysfunction. So the complexes that were affected in presence of dopamine are now reversing dopamine levels. They are also reversed, basically. That was the most important results, really. So here now, we have, at this point, we need to show that actually GST2 occurs also in presence of mutant huntingtin, because in our model we don't have mutant huntingtin, as I told you. And so basically we turned to the rat model to show whether GST2 was preceding the increase of TH, and that's what we show here. So basically, the first one, we show you that BDNF levels, okay? So you can see in panel A that BDNF levels are downregulated at 11 months of age, 8 months of age in these mice, that actually GST2 is upregulated, and that actually TH is still not upregulated, because I showed you these rats, they get basically upregulation of dopamine around 11 months of age. So this was really correlating very well with our model. Now, the next question is whether or not presymptomatic HD actually show a GST2 upregulation. And as you may understand, it's very hard to get presymptomatic brains, because at this stage, actually, they are, thank the Lord, they're still alive and normal. So it's only if they've died for other reasons that there are some brains available. And so basically, we downregulated the analysis, RNA-seq analysis done by other people in brains of presymptomatic HD patients, and we can show basically here that the GST2 is upregulated there too in these patients, and that later on actually is downregulated as in our model. But lately, actually, because there is more of this RNA-seq analysis done on single cells, we have downloaded lots of data of many different kinds of patients, from presymptomatic to HD4, and we actually can confirm even if you divide. So basically, in this one here, so you have control females and control males, and this is actually HD patient females and HD patient males. In both cases, actually, you have upregulation of GST2. So this data start to be very promising to us, that actually possibly a similar situation happens also in patients, which would lead to the fact that then actually one can make some therapeutics which can be used very early on, actually, when the patient still doesn't have all the chorea symptoms, hopefully. Okay, so a conclusion of this is that we believe that chronic loss of neurotrophin signaling in encholinergic neurons leads to the vulnerability of these neurons through the glutathione metabolism dysfunction. And this actually per se leads to dopaminergic dysfunction at the presymptomatic stage, which is then followed by motor dysfunction. So this observation obviously is consistent with the mutant huntingtin that affects cortical BDNF transport or expression and underlie the increased dopamine levels which have been observed in early stage HD. These findings, obviously, that the modulation of the glutathione metabolism pathway, and so basically specifically in encholinergic striatal neurons of mutant tract B, that revert the hyperdopaminergic state and the energy metabolism, blocking basically the early and late motor phenotypes, is very consistent with impaired glutathione homeostasis that can occur early time points, basically. So this one is just to give you the overview of what has happened, so that you don't just listen to me, but so basically, this particular slide says exactly what I just told you. So if you go to the encholinergic cells in green here, you delete tract B signaling, so you have an increase in GST2 that through each DHA function recycle vitamin C that basically, that actually goes to the substantia nigra and increase basically cells which now express actually dopamine that goes back to the striatum. And this causes hyperdopaminergia, striatal vulnerability, and motor defects. So if you actually, through an intracranial injection, downregulate GST2, you have basically normalized dopamine, you have normalized the energy metabolism, you have basically cured the locomotor behavior in these mice. So basically, these results really also support the relevance of this type of models that we have used to elucidate mechanism underlying preferential susceptibility of encephalinergic cells in neurodegenerative disorders such as Huntington. And with this, I just want to acknowledge all the people, especially from my laboratory, that have taken part in this particular study, and all my collaborators, and of course, agencies that have supported me so far. So thank you, and sorry for all these problems with the presentation. Okay, so then it means that I can leave it. It's good.
Muchas gracias. Okay. Recuerde que luego...
¿Dónde has dado?
Luego se tiene que ir de aquí.
Okay.
Luego se tiene que ir.
So we follow with the professor Justo García Yébenes. He, as I told you before, he's the winner prize of 2000 for clinical medicine, the winner prize of the King Jaime I. Then he's been the chief in charge of the neurology department and at the Jiménez Díaz Foundation in Madrid. Also, he is neurology professor at the Autónoma University in Madrid. Also, the director of the Tissues Bank for Neurological Research, and also is the founding president of the Neurological Research Foundation in Madrid. And also he has a biography that I can stay here for the full morning and I never end. So the only thing I have to say is he is one of the, in fact, the best neurologists that there is now in this country, in Spain. So it's a pleasure to introduce you the professor, and it's your time.
Well, thank you very much. It is a very kind presentation. I'm going to talk only about clinical data. So I think that you could be interested in Huntington's disease for a number of reasons. One of that is that it's been a disease who opened one area of research before Huntington's disease. Molecular defects were discovered. There were no suspicions about the mechanism of action, but after that, there are a number of diseases who share the same mechanism, and it could be important to discover treatments for all of these. This is my presentation. I would like some information about previous diseases that could be probably considered compatible with Huntington disease. Then we will talk about George Huntington and his experience. Then we will discuss the clinical features of Huntington's disease. We will talk about the pathology and the genetics. And finally, we will talk a little bit about the available treatments. About George Huntington is considered to be the discoverer of that disease, but there are reports for the last 500 centuries of some cases that could be probably related to Huntington's disease. Some of these cases, were prosecuted, were considered something like witches, something like that, like the patients from Salem in 1692. This slide, there is a painting by Peter Bruegel at the beginning of the 16th century, and there are a couple of subjects that probably have movement disorders compatible with chorea. George Huntington. George Huntington is a New Yorker. His father was also a physician practicing in Long Island, in New York. He used to go with his father to see patients. George Huntington graduated in medicine in Columbia University, and when he finished his medical studies, he went to practice to Ohio. And then when he applied for membership, then at that time he was told that candidates for membership used to deliver a short speech about some medical problem that they have experience on. George Huntington delivered a short presentation of some cases that he has seen with his father in Long Island. And the following years, in 1872, he published a short paper in the Medical Journal of Ohio describing the disease. The description was very short. It was half a page, but he pinpointed the most important findings, such as the pattern of inheritance and such as the spectrum of movement disorders that the patients presented. Yes?
What's the Spanish word for chorea?
Chorea.
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 Thank you.
In general, there are three types of clinical findings in these patients. The best known are the motor features, which include chorea as the most prominent, the most famous, but also other motor findings such as dystonia, myoclonus, motor and vocal tics, Parkinsonism, catatonia, and ataxia. These are the majority of the motor signs. There are also cognitive changes, like lack of memory, disturbance of language, apraxia, agnosia, also behavioral findings such as anxiety, depression, irritability, sleep disturbance. And there are also other findings which do not belong to the central nervous system, but are outside the central nervous system, such as loss of body weight, poor sphincter control, abnormalities of food consumption. In general, the features of Huntington's disease distribute in three phenotypes. This phenotype the first one is the classical variant, the most common, the initial symptoms start from the 3rd to the 5th decade of life, typical motor, cognitive and behavioral clinical features, and progression to death in 15 to 20 years. These individuals, they do have, we will talk about the genetic cause of the disease in a minute, but at the beginning now I can tell you that patients with classical variant, they used to have from 42 to 55 triplets, CAG triplets. There is also another variant, which is the senile variant, which involves probably around 10% of the patients. The initial symptoms are motor and start after age 55. The motor symptoms are characterized mostly by chorea. They do not have, in general, cognitive problems, and they do not have reduction of life expectancy. These patients, they used to have from 39 to 41 CAG triplets. And there is another phenotype, which is the juvenile obesal variant, which also involves around 10% of the population. The symptoms start before age 20 and are characterized by a kinetic rigid syndrome, like a Parkinson's disease. They do have severe cognitive impairment, and they do have rapid progression and death in less than 15 years. These patients, they do have, in general, more than 55. Now, we already went through it. Let's go to the pathology. The pathology is characterized by 3 findings. Finding number 1, which you could see, you see 2 brains. On my side, there is the brain of a patient with Huntington disease. To the other side, there is a normal brain. What you see is an atrophy of certain brain areas, more pronounced in the striatum, especially in the caudate nucleus and in the putamen. The second finding is in the lower part. You see that there are, on the far side from me, there is a loss of neuron. And there is also evidence of positive Huntington staining in the patients, in the brain of patients with Huntington. Genetics. It is very important what could be done by the collaboration of the professionals with the families. In the slide, you could see a family tree of a big family. You can see a picture of Nancy Wessler. She's a psychologist working at the University of Columbia in New York. He used to work, now he's not working anymore. I mean, for a few years. But she built up the Hereditary Disease Foundation, which is an organism which is been funding studies performed in Venezuela, where there is a big family with more than 1,000 cases of Huntington's disease and more than 5,000 cases at risk of the disease. And the gentleman on the other side of the slide is Jim Gusella. Jim Gusella is the chief of genetics in Harvard University. And he was the person who discovered the mutation, which is the expansion of the CAG triplets. This is the spectrum of age at onset with the length of the triplets, and you could see that there is a negative correlation. So subjects, individuals with a small number of triplets, they have a late onset of the disease, while individuals with large amount of triplets, like 65 or 66, something like that, they have a very early age at onset, less than 20. Dr. Minichiello showed this slide, which describes the different molecular events that are produced by the mutation. I'm not going to repeat it, but the only thing is that there are many molecular defects, and it's difficult to say which are the primary one and what are secondary. Perhaps after her presentation, we should pay more attention to the BDNF pathway. We said that there are a negative correlation between the size of expansion and the age at onset, but there are reservations to the mechanistic model. Some of them are genetics and some of them are environmental. Among the genetics, there is a locus in chromosome 15 that plays two independent effects that anticipate or retard the age at onset. There is also a locus in chromosome 8 that anticipates the onset of the disease. And it looks like the studies of association and analysis of metabolic pathway suggest that the genes involved in repair of DNA are some of the modifiers of the disease. I'm going to show you some data related to the environmental modifiers. And this is one study performed by one of my colleagues in Centro Ramón y Cajal in Madrid, is Dr. José Luis López Sendón. What I asked José Luis at a certain time was to evaluate the impact of education on the development of the disease. Why did we do that? Well, there were some evidence from some studies performed in mice with the mutation a few years ago that mice with a mutation that were maintained in an enriched environment, they do have presentation of symptoms later. And also there was evidence that the levels of education had an impact on other neurodegenerative disorders, such as dementia of Alzheimer type and Parkinson's disease. What José Luis did, he had the hypothesis that subjects with higher education, they should have a later development and slower progression. And what he did was he performed one analysis of age at onset and severity in a big registry of data that is in Europe for the last 15 years or so. José Luis started with a database of 4,200, and then after cleaning the table, he ended up with 921 patients. And the variables that he studied was the date at birth, date at onset, date of recruitment, number of CAGs, years of education, age at onset in the father or in the mother, gender, and clinical rating scales, motor, cognitive, conductual, and functional. The independent variable were the years of the school. The database was divided into samples. One sample was this patient who did a study for more than 10 years. In practice, those going to university. And the other group was subjects with less than 10 years of education. In practice, subjects who didn't go to university. Dependent variables, years of onset, motor scores, cognitive score, conductual score, and functional score, and variables of controls, CAG and age. The first data that we have is expressed in this slide, and it was very disappointing, because what we found was that individuals going to the university had earlier instead of later age of at onset of the disease. We don't have any explanation for that. The only one is that individuals going to the university are individuals more capable of detecting minor type of symptoms. So and they are also more likely to attend to physician at an early time. In any case, this is the result obtained. But in the other scores or in the other data, university studies or going to university was able of improving motor score, cognitive score, behavioral scores, and total functional capacity score. The results are here. The motor score going to university improved 3.06 points, which is the equivalent to 1 year of progression of the disease. Also, there were differences in cognitive, conductual, and functional score, and they were also the same. So, going to the university makes you to be able of detecting symptoms earlier, but the severity of these symptoms, motor, cognitive, behavioral, and functional, are better. Now I'm going to talk briefly about the treatment. We cannot cure Huntington's, but we can improve it. And there are a number of drugs that could be used for chorea, dystonia, depression, dementia, irritability, and insomnia. When these drugs are used correctly, the life of the patients would improve significantly, at least for a while. This is one study that I was very happy to conduct in Europe, and this is the study of pridopidine. Pridopidine is a partial is a regulator of dopamine neurotransmission, and it was synthesized and studied from the pharmacological point of view by Professor Arvid Carlsson, my mentor, Nobel Prize in Medicine year 2000. I would like you to pay attention to the graph which is close to me, and that could be the effect of a neuroleptic, a conventional neuroleptic, this drug that is used for the treatment of schizophrenia. Well, so the first part describes the effect of the neuroleptics on dopamine neurotransmission. If your status, excuse me, sorry, if you are not interested, I will appreciate if you could leave the room. Otherwise, you are distracting me. So, if your dopamine neurotransmission is normal, then a classical neuroleptic will decrease it. It also will decrease it if the dopamine neurotransmission is high, and the worst of it, it will decrease dopamine neurotransmission even if its baseline is normal. With predopidine and perhaps with other dopamine antagonists, if your dopamine neurotransmission is low, if there is no dopamine in the synapses, predopidine would act as a partial agonist and would increase activity. If your dopamine neurotransmission is very high, then it would decrease. So for that reason, Professor Carlsson described this compound as a regulator of dopamine neurotransmission. Dopamine stabilizer, able to enhance or inhibit dopamine-dependent function. The effect is through the dopamine T2 receptors. So one study was performed in all Europe. The study was called the Mermaid study because the laboratory, the pharmaceutical laboratory who sponsored the study was in Copenhagen. So it was called the Mermaid, which means multinational European multi-center ACR 16. ACR means Arvid Carlsson Research. Arvid Carlsson, with the money who got from the Nobel Prize, built up a new pharmaceutical company. The study included around 400.
Se metieron en el estudio alrededor de 450 pacientes, de los cuales se pudieron randomizar 437 y se distribuyeron en 3 grupos relativamente homogéneos, uno de placebo con 144, otro tratado con predopidina 45 miligramos, 148 pacientes y otro...
The results of the study were that the patients treated with predopidine 90 milligrams per day, they have 26 weeks after the beginning of the treatment, they have a significant reduction of movement disorders, both if the analysis is of the total, of the full analysis set, or if it was per protocol analysis set. I would like you to pay attention to the fact that the separation of the curve at the end of the study, at N26, is a little bit greater than the separation of the results at 12 weeks and at 8 weeks. But the study was not performed to discover protective roles of pridopidine. The improvement involved the total motor score, which is in the second line, as the modified clinical scale, and produced quite significant difference between the subjects treated with 90 milligrams per day, the subjects treated with 45, and the subject treated with placebo. And when we split the different type of movements, like the eye movements, dystonia, hand movements, gait, and balance, the difference also were significant for all of these abnormalities of movement disorders. Okay, now, there are other treatments for the
Los otros
disease.
tipos de tratamiento. Esta familia que presento aquí, este matrimonio con un hijo, son el primer hijo que se...
That was the first family that we treat with prenatal and pre-implantation diagnosis. Now, the easiest way of ending with Huntington's disease would be eradication. Eradication could be considered with genetic control and with reproductive failure. If that happened in the disease, would happen in patients with large mutations. On the other side, it could be increase in prevalence of the disease if there is a reduction of fertility in the general population, if there is increase of fertility in patients, and if there are new mutations. There are now a number of trials involving genetic engineering, both with oligonucleotides, RNA, and CRISPR. There are a number of studies that have been performed in Europe with tominersen, good results in animal models. These treatments are able of reducing Huntington levels in cerebrospinal fluid, but they are not showing clinical benefits. Dr. Minichiello talked to you about neurotrophic factor pathways. This could be the subject of manipulation. There are also implants of fetal striatum that were very promising at the beginning. Now they are being abandoned. And finally, compounds like pridopidine or compounds which interfere with dopamine pathway could be another alternative. At this point, there is no cure for Huntington's disease, and all efforts should be placed in research and prevention of the disease. Thank you very much.
It's time for a couple of questions. Do you have any questions? Please,
I have a question for Dr. Minichiello. You show us beautifully the involvement of dopamine in the pathology, in the pathogenesis of the disease. In one of the slides that you have shown, you show us that the difference between wild-type animals and not patients, and animals with the mutation, you show us that the difference in the levels of DOPAC were greater than the levels on 3-MT. DOPAC is a metabolite via monoamine oxidase, while 3-methoxytyramine is a metabolite through catechol orthomethyltransferase. Dopamine is considered to be more toxic when it goes to the monoamine oxidase pathway because it generates oxygen peroxide. I think that you have discovered a new pathway for treatment because you could treat the patients with monoamine oxidase inhibitor.
This is fantastic. The point is that it's true what you're saying. Yes, we have seen these differences and we were excited about that, just that we need to show that a little bit more in details to see which one of the two, basically. But yes, it's true. It is so. So it could be another way, actually. But what I'm very fascinated is what you are showing. You support 100% that dopamine is one of the earliest problems in patients. And so basically we need to see how to stop that. So in our case, we just show that the glutathione metabolism can be one pathway that one can start to work on. Because actually, I think it's been seen in juvenile Huntington. Actually, it's been described in juvenile Huntington, but nowhere else, actually. And I believe that one should pursue also this way, because it's an alternative to mutant Huntington per se.
Yeah. One of the problems that we have with this disease is that the phenotype changes with the length of the mutation. So, I mean, at the beginning, or in cases very benign with a short mutation for a long period of time, the caudate nucleus may be more involved and the medium and small spiny neurons may be affected. But when the disease has advanced for many years, or in patients with large mutations, this selectivity is lost. I mean, this is just everything is bad. At the beginning, a group of neurons are affected, but at the end, either temporal end or either mutation end, then everything is involved.
Is exactly this my point, in fact, that actually we cannot look at HD like from stage 2 onwards. Is too late, actually. So one has to go pre-symptomatic. So people with mutation should get basically their genotype and understand how many repeats they have, because this can be correlated with the onset, right, of the disorder. And so basically means that you actually have to go like 10 years or a little bit more before to check, for instance, for this dopamine that is already increased and try to cure pre-symptomatic HD0, HD1, I think. And this would possibly, if we had to use the glutathione metabolism pathway, could be a possibility, which means that you can either delay a lot progression of the disorder or maybe totally I cannot say in humans. In the mouse, yes, it is so. So the mouse does not get any problems ever after we actually have stopped GST02 and reversed dopamine and energy metabolism. So at the moment, our studies are trying more to go to the clinic to try to see in patients whether we can reproduce these early stages similar to the mouse model we have and to the Huntington rat model that is reproducing pretty much many symptoms, including chorea, actually. So that's actually the point. The point is to go early. So it is true what you say.
If there are no more questions, let's give a huge applause.